Let's get in our Zen. You have a class at 9? Okay, great. Anyways, we are going to wrap up quick. But I thought this is a good time to do it early than doing it on Thursday or Friday. How's everyone with biochemistry genetics? All good?
Yeah.
Great.
Need to do more questions, though, see what the second and third level questions are.
Yes. Okay, but for acid-base, do Dr. Panini's cases. They are good. I did a thorough review, I think, but I'm opening question and answer on Thursday session, or we'll plan. Is everyone okay now? Good morning, everyone. Please see that we only had 8 to 9 today, so I'm going to do a quick review on what was left, which is globular protein. Also, yes, please know that if are you struggling with any other concept, let's go to the review. Tell your class reps. So Thursday can be either whiteboard or open Zoom for question and answer. You all do take a vote and decide, okay? Both are either are good for me. Coming Friday also, this Friday also, I'm going to recap again quickly just before the exam. But Thursday, you all decide, okay? Do you want a whiteboard or an open question and answer session? Okay. Okay. Good morning, everyone. So please know you have 14 questions. You have 2 lectures. First, do the lectures thoroughly. Both lectures will be tested equally. You have 2 CV, only 1 CV will be tested. Then do the primers. Remember acid base, I introduced Henderson-Hasselbalch equation and weak acid, weak base drug. We also talked about buffer. You got to know the bicarbonate buffer to know acid-base balance and imbalance. What are we doing today? Amino acid. Please stick to your learning objective. For primer of amino acid, you got to know, are you seeing my slides, everyone? Are you seeing my slides? And can you hear me well?
Yes, we are.
You got to know this predominant non-polar group, please. Everything else is polar. And in polar, there's charge, uncharged. You know base. Base are positively charged or negatively charged. Bases. Bases are proton donor or acceptors. Bases are proton donors or acceptors. They are acceptors. So what charge bases have? Look at the left side. Bases have what charge? Positive. Acids have what charge? Acidic amino acid, negative. Look at the structure. We did amino acid. They have an amino group, carboxylic group. Now tell me which one of them will have a low pKa, carboxylic or amino group? Which one will have a low pKa, carboxylic or amino group? Carboxylic group. Yes, because the acid. Acid, remember, because of acidic nature, the pKa of carboxylic will be low. So usually an amino acid has 2 pKa's, one due to carboxylic, one due to amino, which is constant. Remember, R keeps on changing, which is side chain. That's why this big classification, and you should know the predominant non-polar group. That comes with the side chain. So know that non-polar, will they have charge? Will non-polar have charge? Will non-polar have any charge? No. So what are your charge amino acid? Basic, acidic. Simplest, smallest was, remember, glycine. Later on for your future blocks, not for this one. Please also start accumulating where do you see these amino acid. Did we do glycine and collagen in the last exam? Did we do glycine and collagen in the last exam? Yes. And coming up in the future exam is glycine and purine ring. So there are some places where these amino acids are important. Though you should do first lectures, CV, then primer. But remember these concept of amino acid, that their side chain keeps on changing. That's why they have a classification based on side chain. There's a predominant non-polar group. You got to memorize this group. The rest are all polar. They have only 2 pKa's, but some amino acids have three pKs. Why? One is pKa of carboxylic, other of amino, and the third one is of side chain. When does the pKr comes? Anywhere amino acid have an extra functional group in the side chain. So look at this. There are few amino acids which have an extra functional group on the side chain. They have three pKas. The last one is known as pKr. Naturally, acidic amino acid have an extra negative group in the side chain or acidic group in the side chain. Basic have an extra basic group in the side chain. Cysteine and tyrosine are exceptional. Cysteine also has a functional SH group in the side chain. What does tyrosine have? Do you remember? Does tyrosine has any functional group in the side chain? Anyone? Extra functional group on the side chain? Hydroxyl, very good. So that's why some of the amino acids have three pKas. pKa due to carboxylic, which is constant, amino constant. They have just an extra due to functional group in the side chain. Those three-letter abbreviation is sufficient, but internationally accepted is single-letter abbreviation. And look at this. What can you call this amino acid as? Can you see this box? Should it be, first of all, is it polar, non-polar? Is it polar, non-polar?
Polar.
Polar because it has an extra charge in the side chain. You don't have to identify, is it arginine, lysine? No. Is it basic or is it acidic amino acid? Please don't memorize structure, not at all. Just know the side chain classification and predominant non-polar group. But I always bring all concepts together because you will not pick up biochem again. So is it basic or acidic amino acid structure?
Basic.
It
One
is.
question, Dr. Bach. We will
Yeah.
always assume that we are at body pH, right? To determine
Yes,
whether
absolutely, absolutely, because we are talking all about these amino acid at our body pH. So this is a basic amino acid because look at this pink, there's an extra group in the side chain. Also with the help of the charge, because you know basics are positive, acidics are negative, rest are neutral, they don't have charge actually. You can also calculate a net charge on a peptide. So what's the charge here, net charge here on this amino acid? Plus 1, 2, minus 1. Is it plus 1? Yes?
Yes,
What's the net charge on this amino acid?
is +1.
It is plus 1. Similarly, you can also calculate charge net on peptide just based on this classification. Okay, so keep an eye on this non-polar group. One, that you can understand that each amino acid should have only two pKas. pK of carboxylic should be low because it's acidic. But sometimes some amino acid have an extra functional group in the side chain. This is the group of, this is the amino acids that have three pKas. The extra one is because of functional group in the side chain. And also you can calculate the charge, not just on amino acid, but you can also calculate the charge, net charge on a peptide if you know this side chain classification. Also, did we do that in interactive session? Are there different proteins that are in different environment? Which one is the protein in a polar environment, left or right? Which one is the protein in a polar environment, left or right? Protein, example of a protein in a polar environment, absolutely left. For example, hemoglobin or a hemoglobin in a plasma. Right side is an example of protein in a non-polar environment. This is an example of receptor on the membrane. Now tell me, in this protein left side, which amino acid will predominate on the surface? Polar or non-polar? Which amino acids will predominate the surface? Polar, right? So that is why side chain classification is important for a lot of things. You can know whether this is a chart, this has, looking at the structure, you can know whether it's acidic or basic amino acid. You can calculate the net charge on an amino acid. You can calculate the net charge on peptide. And also you can know how this protein will behave when it's in a different environment, polar versus non-polar. Then we had a primer on protein, which is structural organization. We identified all the bonds and bonds that stabilize. Remember, peptide, we talked about primary structure, we talked about primary, secondary, tertiary, quaternary. Primary structure, we talked about disulfide bond, and sometimes, going back, I'm talking about stabilization. Let's first talk about bond. So there's primary, secondary, tertiary, and quaternary. Primary, we talked about peptide bond, the main bond, who stabilizes disulfide. Secondary structure, we talked about two secondary, alpha helix, beta pleated sheet. We also did the disease. I will tell you first, normal. We talked about alpha helix, beta pleated sheet, interchain hydrogen bond, alpha helix, intrachain hydrogen bond. We talked about tertiary, little folding, gives three-dimensional shape, also function. More bonds come here. Ionic, hydrophobic. Who stabilizes? Hydrophobic. Quaternary, when they say quaternary, that means they are talking about subunit. Now, clinical application. When you talk about primary structure, if primary structure is changed, the whole protein can be changed. Can you give me an example where primary structure is changed? You have learned a disease with hemoglobin where there's a change in the primary structure. Yes,
Sickle cell.
sickle cell hemoglobin. HBS. Yes, you were saying something? HBS, glutamic acid replaced by valine. You have a protein with an entirely different property application. What is it bond? Main bond, peptide bond. Example is insulin where there is disulfide bond that stabilizes them. Secondary structure, we did two disease, the Alzheimer's and Prions disease. So tell me, Alzheimer's or Prions disease, both had beta pleated sheet aggregate, please, which cause the damage. But in which one of them is a conformational change disorder, where because of acquiring an infectious protein, all the alpha helices change into beta pleated sheet? Which one is that? Prions or Alzheimer's? Prions or Alzheimer's? Prions disease, exactly, also known as Jacob's disease. So what are the clinical application of secondary structure? Alzheimer's and Prions disease. Both of them have beta pleated sheet aggregate, but the differences in Alzheimer's, it is because of abnormal cleavage of amyloid precursor protein, whereas Prions disease is quick conformational change of alpha helix to beta pleated sheet. So yes, I will repeat that. What I'm saying is we also talked about different levels of organization and their application, their bonds, primary structure. We talked about peptide and what can stabilize them, insulin, disulfide bond. We also did disease, primary structured loss of change of glutamic acid by valine. You get an entirely different protein with a novel property, sickle cell. So primary structure is important. If it changes, it may bring changes in protein. Then we did secondary structure, alpha helix, beta pleated sheet. Is there what bonds we talk here? Hydrogen bond. There are two diseases we talk here, Alzheimer's and Prions disease, secondary structure. Alzheimer's and Prions both have beta pleated sheet aggregates, but in Alzheimer's, beta pleated sheet are because of abnormal cleavage of amyloid precursor protein, but in Prions disease, beta pleated sheet aggregates result from conversion of alpha helix into all the alpha helix into beta pleated sheet because of acquiring an infectious PRPSC. PRPC is cellular, SC scrapie is pathological. You can acquire, unfortunately, because of number of factors, but both are related to secondary structure. Tertiary, a little more folding, functional, three-dimensional, but more bonds come here. Which bonds stabilize here? Hydrophobic. Quaternary, we talked about subunit. Example, anyone? Example of quaternary? Example of quaternary structure. Hemoglobin, very good. We talked about that. Again, it is also important if subunits are dismembered or subunits disintegrate, even the quaternary structure is lost. This was Prine's disease. It's not because of abnormal cleavage of amyloid precursor protein. It's because of this. See, these are alpha helix. Alpha helix have intra-chain hydrogen bonding. They are wound around only single axis. There's no folding in secondary structure. The actual folding starts in the tertiary structure. And this is beta pleated sheet, parallel and anti-parallel. So this is quick conversion of alpha helix into beta pleated sheet because of acquiring the infectious protein. So the etiopathogenesis of beta pleated sheet aggregate formation is different in Alzheimer's versus Brian's disease. But both are related to the secondary structure or beta pleated sheet aggregates. Beta pleated sheet are secondary structure. Please stick to your learning objective. We talked about globular protein. We talked about hemoglobin versus myoglobin. Try to compare them. Which one is monomeric for storage? Which one is monomeric and for storage and does not have allosteric property and shows hyperbolic curve? Myoglobin, absolutely. We also recognize, please don't forget that give me a second. Please don't forget that we also recognize the term heme protein, where we not just talked about hemoglobin, but cytochromes and catalase. Because of heme, iron should be in a state of Fe+2. So tell me, if iron is in a state of Fe+3, what is that hemoglobin known as and how do you form it? When iron is in a state of Fe+3, that hemoglobin is known as methemoglobin, and you form it by oxidation, not reduction. This reaction keeps going in your body. That's why not just you should know the word heme protein, you should also know the normal hemoglobin derivatives. Methemoglobin is also one of the normal hemoglobin derivatives. What are the other hemoglobin derivatives in your body? Normal hemoglobin derivatives are, what are the other hemoglobin when that are present in your body apart from methemoglobin? Anything else you know? Oxyhemoglobin, deoxyhemoglobin, hemoglobin carrying carbon dioxide, methemoglobin, they are all normal hemoglobin derivative unless they accumulate or they alter pathologically. So you should understand iron only in Fe+2 have the capacity to carry oxygen. When iron is in Fe+3, what does it lose? Capacity to carry oxygen. That's why a lot of methemoglobin means cyanosis, right? Composition you should know. Please know sometimes they write HbA1. HbA1 and HbA1c is not the same. HbA and HbA1 is the same. But they call it HbA. Major adult, minor adult, fetal, and glycated. So tell me, what is fetal made up of? You should know composition. What is fetal made up of? And what is fetal made up of? Alpha 2, alpha 2, gamma 2. Why are these composition example? Now look at this. Major adult is alpha 2, beta 2. Minor adult is alpha 2, delta 2. So tell me, in beta thalassemia, we did hemoglobinopathies. Did we did qualitative and quantitative? Give me example of qualitative hemoglobinopathies. Qualitative hemoglobinopathies. What did we do in qualitative hemoglobinopathies? HPS and HPC, quantitative alpha and beta. Alpha thalassemia means no or low alpha chain. Beta thalassemia means no or low beta chains. So in beta thalassemia, will HbA be high? In beta thalassemia, if you don't know the composition, you will not relate. In beta thalassemia, will the major adult be high? Is major adult high in beta thalassemia? Major adult. So you see, if you're saying yes, you don't know the composition. What is the composition of major adult?
2 alphas and 2 betas.
Alpha 2, beta 2. So in beta thalassemia, will major adult be high? No. Anyone who's saying yes, please correct yourself. In beta thalassemia, major adult will be low. And what will increase other hemoglobin for compensation? HbF, HbA2. Actually, HbA2 estimation is used in clinically to screen beta thal. That is why knowing composition is very important, please. So, no, beta thalassemia, major adult would be low. Which hemoglobin should be high? The other hemoglobins, minor, HbF, HbA2. Yes, they would be high. So unless you know composition, please, you cannot relate clinically. That is where you got to understand the application. See, these are the normal derivatives. But when they accumulate abnormally, especially which one? Methemoglobin, carboxy, sorry, let me go back. What were the normal derivative? Methemoglobin, hemoglobin carrying carbon dioxide, oxy, deoxy. They are normal. Carboxyhemoglobin, carbon monoxide, we will talk about that later, right? Oxy and deoxy. Do you know the difference between oxy and deoxy now? So which is deoxy form, taut or relaxed? Which is deoxy, taut or relaxed?
Taut.
Taut. And when you say deoxyform, remember it does not have oxygen, but what does it have instead of oxygen?
Hydrogen.
Hydrogen ions. When you stabilize deoxyform, are you breaking salt bridges or are you making salt bridges? When you stabilize deoxy or taut form, are you breaking salt bridges? You're making salt bridges. When you are stabilizing deoxy and taut form, so know these terms: oxy relaxed, deoxy taut. Deoxy taut means you are favoring delivery of oxygen to tissue. Also, you can know it as, when I'm saying taut deoxy, is it decreased affinity of hemoglobin for oxygen? When it's taut deoxy, is it decreased affinity of hemoglobin for oxygen? Yes or no? When I'm saying taut or deoxy, is it decreased affinity of hemoglobin for oxygen? Yes. So taut form, let's do only one. Taut, deoxy, decreased affinity of hemoglobin for oxygen. Making of salt bridges. So we got to understand these two forms. Why? Because of oxygen dissociation curve. Oxygen dissociation curve is the curve between percentage saturation and partial pressure. Please don't panic when you see the graphs. Just first see the variables. What does oxygen dissociation curve reflects? Bohr effect. What is Bohr effect? The capacity of hemoglobin, or the function of hemoglobin, to pick oxygen at high partial pressure. Where is high partial pressure? Lungs or tissue? Where is high partial pressure? Lungs or tissue? Where is high partial pressure? Lungs? Lungs, almost 100. And capacity to give up oxygen at low partial pressure, which is 40, which is at the level of tissue. That's known as Bohr effect. Then shape of the curve. Sigmoid is for hemoglobin. Why? Because hemoglobin shows which two properties? Which two properties hemoglobin shows?
Cooperative
Allosteric.
binding.
First allosteric property, due to which there is S-shaped curve. Then cooperative binding. Yes, that's why hemoglobin is S-shaped. Look at myoglobin hyperbolic, right? We talked about that. What is P50, which is down here on the x-axis? P50 is the partial pressure at which hemoglobin is 50% saturated. So now let's see when curve shifts to right or left. That's known as oxygen dissociation curve. This is oxygen dissociation curve, shift to right or left. Okay, what's the difference between positive allosteric, positive cooperativity? And so no. So there are two things. Allosteric means when a ligand binds to the heme, induces a conformational change, which can be positive or negative. What is cooperativity? That when one oxygen binds to one heme, it increases the affinity of other heme for oxygen. So that is cooperativity. They are two different properties. And allosteric can be positive or negative, but cooperativity is positive, right? Allosteric modifier can be positive or negative, but cooperativity is positive. So, and imagine then we do, this is oxygen dissociation curve. If oxygen dissociation curve shifting to the right means you need more oxygen to say reach the same P50. So tell me, shifting ODC to the right, what do you understand? Shifting ODC to the right, you are stabilizing which form? Shifting oxygen dissociation curve towards the right. Are you decreasing affinity of hemoglobin for oxygen when you're shifting the curve towards the right? Well, the shift of curve towards the right, are you decreasing the affinity of hemoglobin for oxygen? Yes. Are you stabilizing taut form? Yes. Are you stabilizing deoxy form? Yes. Are you favoring delivery of oxygen to the tissue? Yes. Normally, it will go on in your body because normally also you need delivery of oxygen to the tissue. There is a slight right and left shift, but when it goes too much, there are factors that entirely push the curve towards the right. What are those factors? Low pH, high PCO2, low PO2, high 2,3-BPG. What's the last one that shift the curve towards the right? High temperature or low temperature? High temperature, hyperthermia. So there are factors that shift the curve towards the right and the left, but you got to understand the implication. Please know that the factors shift the curve, not the other way around. Factors are responsible and outcome is shift to the right. Outcome is, if it's the right outcome, is increased delivery of oxygen to peripheral tissue. Naturally, when will you need them? Metabolic acidosis. Will you shift the curve towards the light? Right. Metabolic acidosis. Yes or no? I'm giving you condition now. In metabolic acidosis, will you shift the curve towards the right? Or will the curve shift to the right? Metabolic
Yes,
acidosis?
you're reducing pH.
Yes. High altitude, when you're going to high altitude, infection, increased viscosity, everything, anything that will the outcome is you want to increase delivery of oxygen to the peripheral tissue. Yes, these factors will shift the curve towards the right. So not just knowing low pH, where you can see low pH, metabolic acidosis. Where can you see hypoxia if you go to high altitude? When can there be hypoxia? Infections. So you got to relate it to the conditions too. You know myoglobin and hemoglobin differences. You know that factors that shift the curve towards the right are high PCO2, low PO2, low pH, high 2,3-BPG, and high temperature. But you got to ask yourself a relevant why and how. You know, low PO2, high PCO2, and low pH all have the same effect. What do they increase? Increase H plus ion, increase ionization, and they make salt bridges. When you make salt bridges, what form you stabilize? When you make salt bridges, what form do you stabilize? When you make salt bridges, you're going wrong. When you make, please, those who are saying R form wrong, when you make salt bridges, you are stabilizing tart form, tart or deoxy form. That's why all these factor by making salt bridges, stabilize deoxy form and decrease affinity of hemoglobin for oxygen, increase delivery for oxygen to peripheral tissue. Also, we did our hemoglobinopathies, qualitative versus quantitative. Quantitative were alpha, no alpha, beta, no beta chains. Where was the change hydrophobic? Sickle cell or HPC? Where was the change hydrophobic in the primary structure? Sickle cell or hydrophobic? Sickle cell or hydrophobic?
Sickle cell.
Sorry. Where was the change hydrophobic? Sickle cell, HBS or HBC? HBS. Now here also, please know, in sickle cell also, it's only the deoxysickle that aggregates and precipitate, not the oxy. So any factor in a sickle cell patient, that's why you should know deoxy, tart, oxy, relax, because even in sickle cell patients, all you got to do, because you can't treat the defect, right? You can't change that primary structure. all the physician tries to do is prevent the factors that will promote deoxysickle form. So tell me, what will you do in a sickle cell patient? What do you want to avoid? Low pH, low PO2, high PCO2, high temperature, infections, going to high altitude. They cannot afford to have more deoxyform, especially deoxysickle. So please know it's very, very important. It's not the oxysickle hemoglobin that aggregates and precipitate. It's only the deoxysickle hemoglobin that aggregates and precipitates. So all the factors that actually shift the curve towards the right are favored or avoided in a sickle cell patient. All the factors that shift the curve towards the right should be favored or avoided, should be avoided in sickle cell patients. They have a lot of painful crisis though. They have to be, they are given analgesic because of occlusion and pain. This sickle cell hemoglobin aggregates, occludes small vessels, but mainstay of their treatment is to avoid any factor that will increase deoxysickle hemoglobin in their body. So see, knowing oxy and deoxy form is so important. Then we did alpha, beta 11, alpha 16. They have related chains. Why these related chains are important? Different stages of hemoglobin development. Embryo have zeta 2, epsilon 2. What does fetal have? Alpha 2, gamma 2. What do adults have? Major, beta 2, minor, delta 2. So the switching on and off keeps going in your body. First, you have an embryonic stage, embryonic hemoglobin switches off. What takes over? Fetus, fetal hemoglobin. And in adult, major adult, minor adult. There is also glycated fetal is very low. Look at this. Look at the second graph. Fetal hemoglobin almost is less, very less 6 months after birth. So why are these two graphs important? Because you got to understand, tell me, in alpha thalassemia or beta thalassemia. There's alpha thalassemia, beta thalassemia. In alpha thalassemia, can gamma chains offer protection in alpha thal fetus? Can gamma chain offer protection here? Can gamma chain offer protection to alpha thal fetus? Or no, they can't. Only in beta thalassemia, gamma chains can offer protection. Why? Alpha rise very early in life. So they are very symptomatic even in fetus. Beta chains rise much later in life. That is why beta thal patients or babies are symptomatic after birth. Why? Who's offering protection? Which chains are offering protection to beta thal fetus? Gamma chains. So understand that alpha rise much early in life, beta rise later. And that is why gamma chain can offer protection if beta chains are low in beta thal fetus. It is not possible in alpha thalassemia. That's why alpha thalassemia actually, because it is because of deletion, there is very low alpha chain. Also because of this loss of protection. If it is very low alpha chain, they have hydrops fetalis, death of fetus in utero. You will see it in alpha thalassemia rather than beta thalassemia. So you got to understand the development of the chains too. So we did these quantitative hemoglobinopathies, sickle cell, HPS, HPC, qualitative, beta thalassemia, quantitative. Please, beta is more common than alpha. What do they have? You see their marrow changes because of anemia. There are marrow changes in beta thalassemia. So, and sickle shape in sickle cell. Sickle shape in sickle cell. And look at this. So this is qualitative, this is quantitative. Beta is more common than alpha, but alpha is also seen. I will tell you different causes in beta and alpha too. But alpha chains rise early in life. That's why fetus may not be protected by gamma chains. Beta chains rise later in life. So fetus may be protected by gamma chains. Look at this. This is major adult, right? And this is sickle cell. Which one is oxy, left or right? Which one is oxyform, left or right? Which one is oxyform, left or right? Anybody? Can you look at this picture and tell me which one is oxy? Yes, left, because oxygen is given up, right? Left is oxy, relaxed, increase affinity of hemoglobin for oxygen. Right is deoxy, taut, decrease affinity of hemoglobin for oxygen. But look at the difference between major adult and sickle cell. Major adult, normal individual, deoxyhemoglobin does not aggregate, precipitate at the base of RBC. They are no, they are also, but they do not precipitate at the base of RBC. But deoxysickle, because of the change, glutamic acid bivalene, hydrophobic patches, sticky patches, they aggregate, they are insoluble, aggregate, precipitate at the base of the RBC. That's why all you do in a sickle cell patient is to avoid deoxyhemoglobin. Beta thalassemia, what is low? Alpha chains or beta chains? Beta thalassemia, what is low? Alpha chains or beta chains? Beta thalassemia, what is low? Alpha or beta? Beta chains. So which chains rise to compensate? There are some tetramers. What are the possible tetramers in beta thalassemia? Alpha 4 and gamma 4. Alpha 4 and gamma 4. Please know that beta thalassemia, will major adult be high or low? There are a lot of ways you can diagnose beta thalassemia. First, looking at the tetramers, then the hemoglobin. Which hemoglobin will be low in beta thalassemia? Major adult or minor adult? Which hemoglobin will be low in beta thalassemia? Major adult or minor adult? Anyone? Beta thalassemia? Major adult, and they also will form some abnormal tetramers like alpha 4 gamma 4. Please see the abnormal tetramers alpha 4 gamma 4 are not much used for diagnosis than major versus minor adult hemoglobin in beta thalassemia, because the cause of mutation in beta thalassemia is splicing mutation rather than deletion, which is in alpha thalassemia. So the amount of chains are much lower in alpha. So the tetramers accumulate much more in alpha, and there they are used as diagnosis rather than beta thalassemia. Right? Beta thalassemia, they measure just the major adult and minor adult. But in alpha thalassemia, yes, tetramers are clinically used for diagnosis. You know, glutamic acid bivalene hydrophobic change is sickle cell, whereas glutamic acid by lysine polar change is HPC, right? And you know the sequence: glutamic acid bivalene, primary structure, hydrophobic sticky patches, aggregate, hemoglobin precipitates at the base of RBC when it's high concentration. Naturally, it's not, it's insoluble, precipitates at the base of RBC, acquiring RBC. This is not hemoglobin, this is RBC. RBC acquire what shape? They acquire sickle shape. And what do you avoid in sickle cell hemoglobin? You avoid all the factors that will promote deoxy sickle hemoglobin. Also, they are given hydroxyurea to increase HbF. So why would you give hydroxyurea to increase HbF in sickle cell patient? Why would you give hydroxyurea to increase HbF in sickle cell patient? Yeah, sure. Sure, I know that. Oh, you got to go to the classroom. Okay, I will finish in 5 minutes. Is that okay? Oh, I didn't realize that you have to go to the classroom. I thought you have a Zoom.
I
Okay.
think we have to go to the classroom. The schedule has been complicated recently.
Okay, so maybe we can do it the rest on Thursday. If you want to go to the classroom, you need time, right?
Yes, if we can do another one, that will be fantastic.
Okay, great. I think let me finish this thalassemia. That's all. Okay. So thalassemia is alpha and beta. Alpha is because of deletion and beta is because of splicing mutation. That's why you see tetramers in alpha that are diagnostic. Tetramers in alpha will be beta 4, gamma 4. Tetramers in beta thalassemia will be alpha 4, gamma 4. Okay, we can do it on Thursday then. We are not left with much. Take care. Okay. Everybody's good?